this video, we will continue to look at the requirements for the growth of microorganisms in the lab setting. We will examine culture media and its classifications based on chemical composition, physical state, and various functional classifications. In order to cultivate microorganisms in the lab, microbiologists must meet the nutritional and physical requirements of the microorganism. Nutritional needs are met by placing the microorganism on or in a nutrient medium. Some microorganisms' needs are more easily met than others. Some microorganisms require specific nutrients and growth factors, making their cultivation more challenging. Most bacteria have not been grown on culture media. There are several reasons for this. Most research regarding microorganisms has been focused on those that are medically important and those that are commercially important. There are some that are medically important that scientists have attempted to grow on nutrient media but have failed to be able to do so. As we examine culture media, we will look at their classifications based on three factors: chemical composition, physical state, and functional purpose. Media can be classified based on their chemical makeup into two types: synthetic or defined media and complex media. Synthetic or defined media contain ingredients of definite chemical composition. In other words, you would basically be able to count the number of atoms of each representative element present in the media. Defined media are used primarily in research because the results of microbial action on the media are comparable between labs, even if they are labs that are thousands of miles away. This is because the chemical makeup of the media is exactly the same. Complex media, on the other hand, contain ingredients, typically plant or animal in origin, that make it impossible to know the exact chemical makeup of the media. Such ingredients are derived from the partial digestion of yeast, beef, soy, or proteins from milk. The partial digestion of such materials releases many different chemicals in a variety of concentrations. Most media utilized for clinical cultures are classified as complex media. Complex media have the advantage of supporting the growth of a wide range of microorganisms because of the variety of chemical nutrients and growth factors available to the microorganisms. Complex media may also have additional ingredients added that provide important growth factors for fastidious microorganisms. Please recall from our discussion of chemical and energy requirements that growth factors are organic molecules that the organism cannot synthesize on its own, and thereby they must be acquired in their diet. Growth factors include such items as amino acids, fatty acids, and vitamins. The media is referred to as an enriched media when ingredients are added to a complex media that allows for additional growth factors. The growth factors are needed by organisms that are fastidious. Fastidious microorganisms can be described as those requiring a relatively large number of growth factors. Culture media may also be classified based on its physical state. The physical state of the media is determined based on the presence or absence of agar. Agar is a solidifying agent derived from the cell walls of certain species of algae. Liquid media are also referred to as broths, milks, or infusions. Broths do not have agar present. Liquid media can be prepared using tubes, flasks, or bottles. Growth in a liquid media can occur throughout the medium and may appear as cloudiness. Semi-solid media have a jelly-like consistency. Agar is used, but in much smaller amounts than is used to prepare solid media. Semi-solid media are useful in assessing the motility of an organism. Solid media utilizes approximately 1.5% agar added to the media to allow it to solidify. Solid media may be prepared in tubes or petri plates. When solid media is placed in a test tube and the tube is placed at an angle while the media cools, a slant will be formed. This increases the surface area for microbial growth. Solid media placed in a tube that is left upright forms what is known as a deep. Each of the three types of media based on physical state play a specific role in the laboratory setting. The storage of microorganisms would be achieved more easily with a broth or a solid media in a test tube. The solid media in the Petri plate allows growth on a solid surface with plenty of surface area, thereby facilitating isolation of a pure culture. Media may also be classified based on their function. Please be aware as we move into this section that one medium may have multiple functions, and that these same media are also classified based on the properties that we have already discussed. To provide an example, one can grow two organisms on a solid, enriched medium that would allow selection and differentiation of these microorganisms. The types of culture media that we will explore based on functional titles are the selective media, differential media, anaerobic media, and transport media. Selective media contains substances that either favor the growth of a particular microorganism or inhibit the growth of unwanted microorganisms. Media may also be made selective if a crucial nutrient is left out of the media. A variety of substances may be used to create a selective media. The addition of the dyes eosin, methylene blue, and crystal violet, and biosalts to media acts to inhibit the growth of gram-positive bacteria. Media to which sodium chloride has been added favors the growth of halophilic or halotolerant bacteria. The image shows an example of selective media. On the left is a nutrient agar plate inoculated with bacteria and fungi. Both organisms grow successfully on a neutral pH of the nutrient agar. The plate on the right, saburo-dextrose agar, is a type of selective media that selects against the growth of bacteria and for fungal growth by using an acidic pH. Differential media are media that allow microbiologists to differentiate among various kinds of microorganisms growing on the same medium because of visible changes that occur in or on the medium. This is possible because bacteria utilize the ingredients of the medium in different ways. Differentiation may be based on a variety of observable changes, including variations in colony size, colony color, changes in media color, formation of gas bubbles, formation of precipitate in the media, as well as other observable differences. The top figure provides an example of differentiation using blood agar as the differential medium. 3 organisms are differentiated on the medium in this image. The basis of the use of this medium as a differential medium is that the microorganisms utilize blood cells differently. The organism to the left is Streptococcus pyogenes, which has the ability to completely digest the red blood cells, leaving behind a clear zone around the bacterial colonies. The bacteria in the center is Streptococcus pneumoniae, which only partially digests red blood cells, producing discoloration in the media around the colonies. And the bacteria on the right, Enterococcus faecalis, does not digest red blood cells, so there are no changes in the media around its bacterial colonies. Another example of differential media is observed in the bottom figure. In this case, the media is known as carbohydrate fermentation media. Carbohydrate fermentation tests are a differential test that determines the ability of an organism to ferment sugars. When organisms carry out fermentation of a carbohydrate, they may produce organic acids and/or gases as end products. Organisms utilize carbohydrates differently depending on the enzymes present within a given organism. Carbohydrate fermentation media contain a specific carbohydrate and a pH indicator within a nutrient medium that provides other required nutrients that support bacterial growth. In addition to the media, a Durham tube, a small test tube, is inverted within the media to serve as a trap for any gases that may be produced during fermentation. In this particular image, phenol red is the pH indicator. Phenol red is a pH indicator that is red at neutral and alkaline pHs. Phenol red turns yellow at slightly acidic pHs. If an organism can metabolize the carbohydrate present, an acid is produced and the indicator turns yellow. If a gas is produced as a result, a bubble will form in the Durham tube. Many media are both selective and differential. An example of such a media is observed in the image. The first Petri plate contains nutrient agar on which both gram-positive Staphylococcus aureus and gram-negative Escherichia coli grow. The middle plate contains McConkey agar to which both organisms were introduced. This demonstrates that McConkey agar is selective for the growth of gram-negative bacteria. This is due to the presence of bile salts and crystal violet. The Petri plate to the right also contains McConkey agar, with 2 gram-negative bacteria growing that can be differentiated based on their ability to ferment lactose. In clinical microbiology, selective and differential media are very important in aiding in the identification of pathogens that cause infections in areas of the body like the respiratory and digestive tracts, where microorganisms normally reside as part of the normal flora. Specimens from such infections contain the pathogen along with bacteria of the normal flora. Selective and differential media aid in the isolation and identification of a pathogen. When cultivating anaerobic microorganisms, the microbiologist must successfully create an anaerobic environment. One method utilized by microbiologists to create an anaerobic environment is to use a reducing media. Reducing media contains sodium thioglycolate, which is a substance that binds to oxygen and reduces it to water. The media also contains an indicator, methylene blue or resazurin, which shows the location of oxygen by changing the media either blue or pink respectively. A small amount of agar is added to the media, making it very thick. When the sample is autoclaved, oxygen is driven out of the sample, and as it cools, oxygen diffuses into the media from top to bottom, forming an oxygen gradient. Another method of creating an anaerobic environment is to utilize an anaerobe jar. Anaerobe jars are containers with tight-fitting lids that prevent oxygen from entering. After the inoculated plates are placed in the jar, water is added to a full packet, and the lid is placed on the jar. The water reacts with the chemicals in the packet to produce hydrogen and carbon dioxide. A palladium catalyst then reacts with hydrogen and oxygen to form water. A paper strip impregnated with methylene blue is placed in the jar as a visual indicator. The blue color of the dye is only visible when oxygen is present to oxidize the dye. Transport media are media that contain salts and buffers necessary to keep the organisms alive and maintain their ratios for a short period of time until they can be transported to the lab for identification. Hospital personnel are responsible for properly taking the sample and for using the proper transport media. Both aerobic and anaerobic transport media are available for clinical specimens.